We already talked about this in class, but I just wanted to kind of remind you some of the most important cell types are in this slide. That would be the mucus cells, parietal cells, chief cells, G cells, and D cells. The D cells are not shown here. And remember, this is actually kind of like your universal gastric pit and universal gastric gland. In reality, some of these cell types are more prominent in different parts of the stomach. Some of them are more prominent in other parts. So, for instance, like I said in class, the parietal cells are actually more prominent in the body and fundus of the stomach, which would be the middle and then the top of the stomach, respectively. So, again, this is just to kind of show you all in one kind of image the different cell types. But again, these have different proportions depending on where you are in the stomach. Okay, so again, we already covered all this, but what I'm going to do is get into this in much greater depth, especially the parietal cells, the D cells and the G cells, and really a lot of these. And there's at least one more cell type that I'll mention that kind of runs behind the scenes, not directly in the lumen, called the ECL cell. We'll get into that in a little bit. So I'm going to go ahead and bypass the animation that we went over in class. And sorry about that. Bypass that animation. And we're going to go just briefly through this table. We already kind of talked about this to some extent. Just wanted to remind you of how gastric acid production is regulated in the stomach. Remember, there are three phases: the cephalic, the gastric phase, and the intestinal phase. The cephalic phase is, again, it's based on, it's the central nervous system. It's actually the thought processes, the sensation of food through smell, even the taste of food, just thinking about food. Believe it or not, these things are going to trigger the parasympathetic nervous system. That activates the enteric nervous system, which is in the entire GI tract, and that will trigger acetylcholine release onto the enteric nervous system, and that actually increases gastric acid secretion. So that's just even before the food is. I mean, it doesn't have to be in your body at that point. It could be just the smell of it. Gastric contents would trigger the gastric phase. That's through swelling or distension, protein or peptides. And your stomach actually responds where it has, I want to clarify, this is low protons, low acid. That means high pH. So if the stomach gets too alkaline, it has an automatic switch to produce more acid. So that's regardless of the food content or distension, just to maintain the correct acid concentration. And again, that would be through long and short neural reflexes and direct stimulation of gastrin secretion. We'll talk about those. Intestinal contents, and I like to be as logical as possible, to try to look at things in a logical manner rather than memorizing every detail. So the question I would ask then is, once the chyme or the nutrients and fluid reaches the small intestine, what should happen? What should the pH be? And basically we know the pH in the small intestine should be neutral, or it could be slightly acidic, neutral, or slightly alkaline, but regardless, it's much more alkaline than the stomach in the duodenum. So the first thing I wanted to focus on is then the proton concentration. With high proton or high acid, that's a problem. The small intestine shouldn't have that, like I said. So that will actually trigger to the stomach to actually stop HCl secretion at that point. So it's basically pretty simple. So when the small intestine has too much acid, it wants to correct for that, and it basically triggers less HCl secretion from the stomach. So it's the small intestine's way of telling the stomach to slow down. You've produced enough acid. The small intestine says, well, I already have enough acid, and actually maybe too much, so I'm going to tell the stomach to stop producing acid while I do my job. And the duodenum's job is to receive the pancreatic enzymes and the bile from the liver, going through the the bile duct and the bile acids coming in, and all that together is going to start to mix the food with the digestive enzymes and start to digest the food, and then it travels to the jejunum where the real thorough digestion occurs. But all that has to occur at a close to neutral pH, definitely not very acidic pH. Some other things would be distension in the small intestine. Again, it's just logic. So that means the small intestine is filled with food. It's going to be distended or swollen. So it tells the stomach, well, you got to stop digesting food for a little while and let me do my job, right? Until you break down more food and send it my way, right? Because when the food gets broken down into smaller particles, then it can travel through the pyloric sphincter. So it's going to stop the stomach from doing that for a period of time until the small intestine is empty, or at least the nutrients are gone. And that's the same. The same holds true for all this: nutrients, osmolarity, anything that is symbolizing or representing the intestine not being finished with the food digestion process. It's going to slow down the HCl secretion in the stomach at that point. Now, I hope this is a kind of a good thing, but I actually have a few different slides showing something very similar from different perspectives. And what this is an image of the main acid-secreting cell in the stomach, and really, as I mentioned in class, the parietal cell. Right outside the parietal cell, we have the highest concentration of acid that you'll find in the body. That's where protons get directly secreted by the proton pump. So this is the simplified version. So I want to start here, and the most important thing to know again is first of all is the main functions of each one of these things, and which ones trigger acid and which ones suppress acid. So we kind of have this trio or trifecta of acid secreting substances. One of them is gastrin, and that is a hormone that's pro-acid. That's why it's a positive there. Histamine is pro-acid. That's why it's a positive. And acetylcholine is pro-acid. Now, acetylcholine comes from the actual parasympathetic nervous system, the vagus nerve. So that's where you get your smelling food, thinking about food, tasting food. That will trigger acid secretion, and some other things that we'll talk about. Whereas gastrin and histamine are much more responsive to the local environment. So is there food in the stomach? And that food in the stomach will trigger these histamine and gastrin to be released to trigger acid secretion. So that's the gastrin and histamine are much more local. The acetylcholine is much more central. Somatostatin is like our checks and balances. That will be our opposing guy, like the yin and the yang. So somatostatin would be, we'll say, the yang, or whichever one you choose, but it's opposing. So the somatostatin is actually going to oppose acid secretion. And now this diagram has it binding directly to the parietal cell. A lot of the references that I've used show that it actually binds not directly to the parietal cell, but to a different cell that I'll talk about. I don't want to focus too much on that, especially for testing purposes, because it's kind of a nuance. So again, this is kind of a reiteration of that. It's a little bit more advanced. So we have, we can start at the top or start at the bottom. But if we start here, the cephalic phase is, again, that's your smelling food, your tasting food. That's coming from the central nervous system, the brain, and that will cause acetylcholine release here, here, and here. So that now I said that gastrin and histamine are more local, but it doesn't mean that they can't be triggered by the parasympathetic nervous system. But what I mean is that gastrin and histamine, they can actually function and be released even without the parasympathetic nervous system. So they can be released on their own due to local responses. Whereas acetylcholine really has to be released through the central nervous system, the parasympathetic branch of the autonomic nervous system. So acetylcholine causes gastrin secretion, it causes histamine secretion, but it directly causes acid secretion. Okay, and if we look down here, I don't want us to miss this. That would be rather than just thinking and smelling food, thinking about it and smelling it, we have, if we actually have food, especially distension of the stomach, amino acids and/or peptides, if we start here, that'll actually directly trigger gastrin secretion, as I said. So that gastrin can be released through this direct local mechanism, through food in the stomach, or through the sensation or thinking of food and smelling food. Okay, and gastrin is a huge trigger for acid secretion as well as histamine. Okay, and remember, somatostatin is the good guy in the sense that if we want to block acid secretion, we want more somatostatin activity. I have to warn you, this is a kind of busy, complicated-looking image, but it's something that I actually made, and I'm going to explain it. But if you want a kind of more thorough explanation, I actually posted this on Wikipedia, and actually there were two images on Wikipedia related to gastric acid secretion, and actually somebody deleted the other one the one that I didn't make, and it wasn't me, believe it or not. So now this is the only image on the page for gastric acid secretion. So I'm going to pause it for a second and bring up that page so I can show you what I'm talking about. Okay, so we're on Wikipedia. If you type in gastric acid, you'll find a lot of information. Okay, but I added this image right here, and actually I would say the more detailed and more focused explanation of the image would be if you click on it, scroll down, you see my name there. You scroll down though, and you have a full description of what this is explaining. Now, some of this gets into the drug mechanisms, which you do not have to know those for the physiology course for the test. But if you start at the top here and look through everything, it really explains everything, the mechanism and the way everything on this diagram is kind of interacting. So I'll walk you through it. But again, if you miss something, just go through this and see if you could find the explanation that you need there to make more sense out of it. Otherwise, you can ask me on the discussion board. So I'm going to move back now to the slideshow. Okay, so we're back on the slideshow, and so what do we do with this overwhelming, gigantic beast of an image? Well, it looks bad, but we can actually break it down, and please kind of trust that I'm going to do that, and kind of take it a piece at a time to make sense out of it. First of all, I have to clarify the right side of this is meant to represent the bottom of the stomach. The left side is representing, actually this is fundus, so that would be the top. It could also be the body, okay, which is the middle of the stomach. But I want you to understand there's a physical separation where a lot of the G cells and D cells are going to be, and this would be in a gastric pit. You don't see the gastric pit here, but so I want you to envision, we already talked about those. See, the G cells and D cells are together in a gastric pit, and remember, they can release things actually into the blood. And if we look, down the bottom here, you'll see we have proteins and we have acid. So the bottom here is supposed to be, this whole thing is supposed to be the lumen. Right here and here, this whole thing is supposed to be the lumen of the stomach. That means where the food or chyme is. Back here is the basolateral surface of the cells in those gastric pits that we talked about, okay, just a little while ago and in the last class. So first I just wanted to kind of orient you, and hopefully that makes sense, that you understand kind of physically where we are in the stomach. And then we'll go ahead and start talking about what is actually happening. So first of all, before I miss this or accidentally skip it, I wanted to talk about the central nervous system part of this before I get into the local mechanisms. So this is representing the parasympathetic nervous system. Remember, it's releasing acetylcholine. So if you're smelling food, tasting food, you'll get a pretty early trigger, pretty quick trigger. That it's a positive trigger for, first of all, a cell type that we haven't really talked about it yet, but we're going to do that in a little bit. That is called the enterochromaffin-like cell, or enterochromaffin-like cell, or ECL cell, which releases histamine. That's as far as we're concerned in our course. We're just going to talk about that. It may have other roles. The main role for our purposes is a histamine release. Histamine is pro-acid. So this is kind of a roundabout way, but acetylcholine triggers the muscarinic. You don't have to memorize the specific receptor, so don't worry about that. But it triggers the ECL cell to release histamine. Histamine, and you do need to know this, is the H2 receptor, because in pharmacotherapy you'll learn about the drugs later on. The H2 receptor is activated by histamine to trigger acid secretion. In this parietal cell, remember, that is the main acid secreting cell. Now, I'm not showing it here as the way it's normally depicted, which is having channels on the left and right that are kind of like pathways for the protons to flow. So I kind of simplified it. So that would be more accurate. This is a little simplified. But all you need to understand is that the histamine is triggering the proton secretion. Okay. So what else does acetylcholine do? It also triggers directly the parietal cell. So it's indirect triggering of the parietal cell to secrete protons. Okay. However, that wouldn't be very smart for the body to do without some counter-regulatory or protective mechanism. So, this is a physiological process. If it were always detrimental, something would have been wrong, right, with the physiology and really evolution. So something here has to be positive, not just pro-acid. And acid is good, right? If you're talking about digesting food and talking about protecting from microorganisms, but we don't want the acid to be able to directly attack our other cell types. So what will happen is simultaneously acetylcholine will trigger a mucosal epithelial cell. So it's not parietal, it's a mucosal epithelial cell, and that'll secrete mucus and bicarbonate. The mucus creates a thick barrier so the acid can't attack this cell type. The bicarbonate neutralizes any acid that starts to penetrate into the mucus. Okay, so we have a kind of a balance between pro-acid and antacid, and the pro-acid allows food to get digested, whereas the antacid protects these other cell types. Since it's right here, I might as well talk about it. The prostaglandins E2 and I2, we've talked about those before as vasodilatory prostaglandins causing vasodilation, but they have other functions, and one of those is protection. Prostaglandins E2 and I2, they stimulate the mucosal cells to produce mucus and bicarbonate for protection, but they inhibit the parietal cell to essentially block acid production. So that is something that can be regulated or fluctuate up and down depending on needs. So if, as you could probably already imagine, if you remember anything about the medications, well, I should apologize because you haven't learned about them yet, but you may have heard that some medications like NSAIDs block prostaglandin production. Just because NSAIDs are such a popular drug class, you may have heard of that anyway. But this enzyme cyclooxygenase 1, which is the target of NSAIDs, this enzyme is necessary for PGE2 and PGI2 to be produced. So if you block that with a drug like an NSAID, that would actually block this protective effect, because you wouldn't have enough PGE2 and PGI2. Now, so that was more, again, that's just the cephalic phase. Actually, we're just mostly talking about just when the food is just being smelled or tasted. But what happens when the food is actually in the stomach? So I'm showing, obviously hot dogs have a lot of other things other than protein, but just to make it kind of simplified, just showing the hot dog as a kind of representing the meat or the protein, and that is actually one of the major triggers for the G cell. The G cell is what secretes gastrin, and gastrin here I'm just making just to make it easier to understand and recognize. It's kind of like the green toxic little bubbles, and so basically, because from the context of pharmacy, we're generally talking about acid being something we want to block rather than stimulate. So, apologize for the gastrin fans out there, but basically we're going to call him the bad guy here. So gastrin travels, and again, this is kind of like a microendocrine or a paracrine system. So the gastrin will travel through the bloodstream, right? And this is behind. This is not in the lumen. This is behind the cells in the gastric gland or pit. So gastrin is traveling, and then it'll diffuse out of some capillaries. It'll stimulate the ECL cell to produce histamine. It'll also directly stimulate the parietal cell to produce acid. So gastrin is direct and indirect, just like acetylcholine, with respect to the ECL cell in the parietal cell. So gastrin is not really good to have excess of. Now I put somatostatin, which is kind of our hero here. Somatostatin is kind of like soothing blue, like cooling effect. As you notice, you probably noticed I color coded a lot of things, and red generally means pro-acid here, and blue generally means protective or antacid, and that holds true with the D cell and somatostatin as well. So how does the D cell work? It's pretty intricate how the body is designed to do this, but what will happen is, just say for instance the stomach made too much acid, particularly if the pH goes below 3. That's a lot of protons or a lot of acid. That creates a negative feedback effect. That directly triggers the D cell. The D cell secretes somatostatin, and it has two effects. One of them is and I apologize because I called gastrin more of a paracrine effect, but it's actually endocrine because gastrin does travel to some extent. Whereas this is a true paracrine effect, whereas the D cell releases somatostatin, right? That'll travel pretty locally to a G cell to block it directly. So the more somatostatin release, the less gastrin release. So they're kind of fighting against each other, and somatostatin blocks that. However, somatostatin also travels. Apologize for the accidental vulgarity. Somatostatin also travels through the endocrine pathway, and that can directly bind to the ECL cell to block histamine release. Okay, so again, somatostatin then is going to be protective, gastrin is going to be pro-acid, so we need an appropriate balance so the stomach doesn't have too much acid. So I don't want you to really memorize every receptor type. I do want you to know the H2 receptor. And for gastrin, you can just call it the gastrin receptor. And somatostatin, you can call it the somatostatin receptor for the most part, for now, okay, until you get to pharmacotherapy. So I wouldn't worry about memorizing too much about the actual names. I do want you to understand the H2 receptor, though. So now, if you were curious, you could kind of investigate this box and kind of understand what all the drugs are doing. At least some of them are numbered on here on the diagram itself. Some of them don't work on directly on a cell type. Some of them actually just work in the lumen of the GI tract. So I'm not going to talk about the drugs, but if you're curious, you can see all those targets there. And that'll be held for next year in pharmacotherapy. Okay, so I just have a simple question here based on what we were talking about. A little bit of recall from the previous lecture as well. An increase in protons in the small intestine affects secretin production in the small intestine and acid production in the stomach. In which ways? Or in what ways? So remember, secretin, okay, this was actually in the vocabulary, so I hope you've gone through that. But secretin is produced in the small intestine when there is basically, if there's a lower pH or there's acid getting in. Again, if there's acid getting into the duodenum or distension, secretin will travel as an endocrine effect. It'll travel to the stomach and actually reduce acid secretion. So basically, if there's too many protons in the small intestine or it's too acidic, secretin secretion will go up or increase, which is A, and gastric acid secretion should decrease. So let's clarify that. So the first part of this is correct. The second part is wrong. B would be correct because secretin secretion increases and gastric acid secretion is reduced. Okay, so the purpose of secretin is to slow down acid secretion in the stomach, and that's primarily for the small intestine to not become too acidic and for the small intestine to have time to digest food before the stomach breaks down more food, essentially. Okay, and the rest of this is really everything that I talked about. I should say at least several slides here are what I just talked about, and it's really directly related to that image that we just discussed. So, enterochromaffin-like cells, please understand their function, how they're affected by acetylcholine, gastrin, and somatostatin, and the fact that their main function is to release histamine, and that is pro-acid. So please understand all that. So there's balancing effects, positive and negative, on that. The parietal cells, remember that, so you have to know that's the main cell type that secretes acid. Also understand the main triggers. So acetylcholine is pro-acid there, histamine is pro-acid, gastrin is pro-acid. Okay, the only thing that is antacid that's really directly affecting parietal cells would be the prostaglandins E2 and I2 on the EP3 prostaglandin receptor. Okay, so there's not a lot of direct negative effects to suppress acid there. There's mostly positive. Okay, and that's actually right here. So E2 and I2. We just talked about that. So I'm going to go ahead and move on to actual stomach motility. I want to actually talk a lot about stomach motility. I do want to talk about gastric emptying and what controls that. And then, when we talk about motility, it'll almost all be the small intestinal motility and how peristalsis occurs. So again, not too much. We don't have a lot of drugs that at least are intended to directly affect stomach motility specifically. So I do want to talk, like I said, gastric emptying is what I wanted to kind of focus on. So first of all, what does that mean? Okay, if it's a term you've never heard before, I want to kind of clarify. Gastric emptying is just when the stomach empties its contents into the duodenum, so which is the beginning of the small intestine. So it's very different from, for instance, a bowel movement. Okay, so gastric emptying is sorry, I thought I hit the mute button, but accidentally. But it looks like I didn't. Just change the volume. Apologize for that. So, so what we're talking about is, so what actually triggers the stomach to empty its contents in the small intestine? So this is simple, kind of logistically or logically. It's just that the stomach will empty its contents, first of all, when the food has been digested enough, and second of all, at least as importantly, when the small intestine is ready for it. And that means when the small intestine has processed all of its food that was prior to that or previously in there, the food is processed, it's moved on, and the small intestine is empty in the sense that it's finished digesting and it's already passed the food on to the next section of the small intestine. Okay, so if we start here, you can see how that works. So we're starting with the duodenum. The duodenum is pretty much the control point there. So all these things, which would be high acid, and rather than mentioning every single thing, it's just high acid, and then the presence of food or nutrients putting pressure on the stomach. Pressure on the small intestine or the duodenum. So that tells the duodenum that, okay, so there's a meal coming, or there's a meal here. We need to buy some time to process it before we get more. So we're going to want to slow down gastric emptying if there's food there. So a couple of things can happen. We could have secretion of things called enterogastrones, which are hormones that'll travel into the plasma, and that doesn't have to travel very far. It'll travel right to the stomach, okay, from the small intestinal blood supply to the stomach. So again, this is not in the lumen, right? This is not where the food is. We're actually getting hormones traveling in the bloodstream, kind of behind the scenes there, traveling to the stomach to slow down gastric emptying. Also, we have neurons there, which are part of the enteric nervous system, that are triggering signals to the central nervous system. The signal might be, I'm not hungry anymore. If there's food in your small intestine, the CNS might say, you don't need to eat right now because you already have food in your small intestine. So that's a much higher level effect. Whereas, and again, that's the kind of afferent pathway. Also, part of the afferent pathway or the sensory pathway would be um, going to the stomach to stop gastric emptying. Okay, like I said. So we have two different pathways: a long pathway going to the central nervous system, and a short pathway going to the stomach, and that's just the neurons. So this right side here is purely neuronal. That means there actually is a nervous system embedded in the GI tract that's communicating with different parts of the GI tract, and one of them is duodenum to stomach to slow it down. Okay, so that, like I said in class, some people consider the GI tract like a second brain because it is, I think I read as many neurons as a spinal cord or very similar to that. So it's highly innervated. So I just want to remind you, the sequence of this is based on the sequence of, gastrointestinal contents moving from the mouth all the way to the anus throughout the entire GI tract. So we're going in that sequence. So the next structure that's going to be important is the small intestine. Okay, I'll spend quite a good amount of time on this because it's so critical. The small intestine is immediately after the stomach. It has, in order, the duodenum. Some people pronounce it duodenum. I prefer duodenum, jejunum, and ileum. The small intestine is the most, definitely the most important absorptive portion of the GI tract. That's where fats, proteins, and sugars are absorbed. So, if you lost a large section of your small intestine, you would definitely have impaired absorption of some nutrients. Okay, and a lot of other things. So the duodenum communicates with other GI organs and receives secretions from the liver and the pancreas. That's from the hepatopancreatic ampulla, where the pancreatic juices and the liver bile actually would merge, and we'll talk about that again. The jejunum performs most of the absorption through the small intestine. The jejunum is very, very long. The duodenum is very short. The duodenum is just, I think of that as like that's your receiving vessel. So you're receiving gastric contents in the duodenum, you're receiving bile, you're receiving pancreatic juices, and that's kind of like where it just starts to kind of all come together and mix. But then when it moves on to the jejunum, it's a very, very long tube where constant mixing and absorption occurs. So it has to be very long to give it kind of a long time for absorption to occur, to pretty much have 100% nutrient absorption would really be the goal if the jejunum had one. So again, the duodenum is only about 10 inches long. The jejunum is about 8.2 feet long, much, much longer. Okay, so like I said, everything is happening here. There's digestion of duodenal chyme, that is enriched with trypsin, which is a protease, amylase, which is breaking down starches, and bile, which is emulsifying the fats. I strongly encourage you to review what an emulsifier does. We talked about that in class. I definitely plan on asking on the test what emulsification means. And so what is, rather than just saying bile digests lipids, that's technically not accurate. It emulsifies lipids, and digestion occurs through lipase, which is different from bile. We'll talk about that a little bit more later. Okay, and most of the nutrient and drug absorption occurs in the jejunum. So you can again imagine if someone had a bowel resection, if part of their jejunum had to be removed through, because they had ulceration, such as ulcerative colitis or Crohn's disease. Actually, ulcerative colitis is the colon, so I apologize, but Crohn's disease could be affecting the small intestine, causing ulceration. And then the ileum is also pretty long. It doesn't do as much absorption as the jejunum. It does absorb some things, which I don't want to get into because it's a little bit too much detail. But at the end of the jejunum, we have the ileocecal valve. It's kind of similar to the rest of the GI tract, where prior to the stomach we have the lower esophageal sphincter. the end of the stomach, we have the pyloric sphincter. At the end of the small intestines, we have the ileocecal valve, which it physically isolates intestinal contents from, at least small intestinal contents, from large intestine contents. So from small intestine to colon, there's a separate, kind of like it is a valve in a sense. It's a very small pathway that has to, well, it's small until it dilates to allow fecal matter to enter. Okay, so that's physical separation there. So probably easier just look at the image when we talk about these things. So duodenum, quite short. Jejunum is pretty long. Ileum is pretty long. So it's very, very dense and bundled into a tight package, but it's actually very, very long, as we just talked about. So this is where we transfer from small intestine to large intestine, where, of course, it gets larger, and it's structurally quite different. And this is where we find the appendix. Okay, and then we have, which we'll talk about this a little bit more in more detail later on, but we have ascending, transverse, descending, and sigmoid colon. Okay, so that's from start to finish in the intestines. So just a little warning, we're getting into a little bit more complex, a little bit more detail, and a little bit more complexities in the small intestine right now, and how the small intestinal musculature works to not only move food or chyme forward in the caudal direction, that's caudal, and also to mix food to expose more surface area. So just this is, and again, I don't know if I mentioned this before, but I usually mention it in several courses. I would say Nature Reviews is probably one of my most favorite places to get kind of like a very nice summary of information on any topic. This one was neuroscience, even though we're talking about the GI tract, because the nervous system is so prominent in the GI tract. I found this actually in the neuroscience journal or the neuroscience section of Nature Reviews. Nature Reviews are awesome in terms of diagrams and kind of distilling it down to a user-friendly, digestible format. So I strongly encourage that for really any topic. So back to the topic at hand. So this would be the lumen of the small intestine. It has a lot of surface area. If we go a little deeper, we have the submucosal plexus. That is a network of neurons. Submucosal plexus, that mostly controls secretions of fluid and enzymes. Okay, whereas the myenteric plexus, and again, this is not as black and white as I'm making it seem to be. These nerve plexuses are actually connected to each other, as you know, although you can't see it here. They're all connected, and both of these plexuses are connected to the central nervous system. So it's much more complex than it looks here. But the myenteric plexus is in between let's look all the way over here, in between the circular muscle and the longitudinal muscle, whereas circular is like, really it's a continuous sphincter throughout the entire intestinal tract, which is circular muscle, just like your iris in the eye, which kind of can constrict or dilate. Whereas the longitudinal muscle is kind of like more like if you look at a biceps type of muscle where it's elongated, and this is going perpendicular to the circular muscle, so it's a different direction, and that has its own role as well. And they really have to work together to move food in the proper direction. I don't have a lot to say about this diagram, but I just thought it was a little bit easier to understand the layers. Going from outside in, we have the serosa longitudinal, as you can see, perpendicular striations to the circular muscle. A nerve plexus, that's the myenteric plexus. Submucosal plexus has a lot more blood supply, and it has its own neurons. But the submucosal region has a very large blood supply. Okay, so when you're thinking about, okay, I tell you food is absorbed, drugs are absorbed from the small intestine, but what does it get absorbed into? And what happens is it actually gets absorbed into the intestinal cells, but then right next to those you'll actually have capillaries in a lot of the villi, okay, which are branches from these vessels, and that'll travel right into the veins or venules, and then it'll travel actually right into the portal vein and right to the liver. So just to see if you guys are kind of sharp and alert, just a reasoning and recall question. So NSAIDs are drugs that inhibit prostaglandin production. If NSAIDs act on the stomach in this way, what would be their expected effect? Right, and I already kind of alluded to it. Remember, they block prostaglandin E2 and prostaglandin I2 production. Those are protective prostaglandins. So NSAIDs would actually decrease mucus and decrease bicarbonate secretion. So those are two ways in which NSAIDs could be placing someone at risk for an ulcer, because they're thinning the layer of mucus and they're decreasing the bicarbonate, which neutralizes the acid. So that's why those drugs, such as ibuprofen, indomethacin, naproxen, ketorolac, there's so many of them. They all have different levels of risk, but they all inhibit prostaglandin production, and they all thin the mucus in the stomach, and that's why a lot of people don't realize that. That's why even if you had an IV NSAID, even if the NSAID's already in the blood, that's the risk is there. The risk actually comes more from that than it does from the NSAID being in the stomach. It comes more from the NSAID being in the blood, traveling to basically behind the cells of the stomach, on the basolateral side, and blocking prostaglandin production. So we're going to talk about regulation of the small intestinal muscle motion. So smooth muscle, secretions, and the role of the neurons. As I mentioned, there's two muscle layers that we talked about. The small intestine secretes various substances into the lumen. That would be mucus. Various substances in general, but mucus into the lumen, chemicals into the blood, such as cholecystokinin or CCK. And I would say cholecystokinin is probably, out of all the chemicals that the small intestine secretes into the blood, CCK is the one that I really want you to know very, very well. It's one of the main ones. Okay, we talked about secretin. I want you to know that. Now I want you to understand CCK, or cholecystokinin. So a lot of times I notice that students get confused about, okay, they might understand what cholecystokinin does, okay, but then I ask, okay, what causes it to be released? What organ does it come from? What organ does it travel to? And then there's a lot of confusion. So I want you to understand that cholecystokinin is made in the small intestine, okay? So it's released into the blood from the small intestine and travels to the pancreas, okay, and some other organs that we'll talk about. But it starts off in the small intestine and travels from there. So, and hopefully that makes more sense when you think about the fact that the cholecystokinin is released when there is food in the small intestine. So that's the small intestine's way of communicating. That's one of its ways of communicating. Okay, so we're going to get into that in more detail in a little bit. Just back to one concept before I get into those details. That again, the small intestine has different ways of interacting with chyme. Right, it can, and this way is one of them that I wanted to kind of highlight, and that is the segmentation or mixing motions. Okay, so if you look at this, you could see the arrows are going in both directions. What this diagram is showing you is that if the small intestine were just mixing, the food would not necessarily go in the correct direction. The net motion of the food would go, in either direction. So I guess the question then is, what makes food go right in the correct direction, right, to be defecated? And that's going to require not just this mixing motion; it's also going to require peristalsis. So, and it's not just circular muscle that's used in peristalsis, but circular muscle is critical there. Okay, so this mixing motion, a lot of that is longitudinal muscle. A lot of the peristalsis is circular muscle, but really they work together. So I don't want you to worry too much about what muscle type is working here. I want you to be more, just more aware of the difference between the segmentation, which is mixing, so the segmentation type of motion, and then the peristaltic type of motion. Okay, we need really need both of those. So back onto the just a reminder basically of the nervous system and the structure of the intestinal wall. So I already showed you kind of the layers, so I just wanted to reiterate what that looks like. If we're starting we can start from the outside. The lumen of the small intestine, where the chyme is, we have the intestinal cells here. Okay, some of those are endocrine, right? Some of those, that means that the endocrine secrete substances inside, and that'll reach the bloodstream. Exocrine secretes things outside, right? And that will be right in the chyme, right in the lumina of the small intestine. Okay, so we have different cell types. We have several muscle layers. The muscularis mucosa, which we didn't talk about, that's actually not well understood. Some scientists in the field believe that it vibrates, and physiologists, that it vibrates at a high frequency to maximize diffusion of nutrients. Again, that's not well understood or confirmed. The purpose of that, at least. Circular muscle, as you're really just seeing the ends of the muscle fibers because these wrap around, and then longitudinal muscle. And remember, we have the submucosal plexus and then the myenteric plexus, and these really work together with the central nervous system. Okay, so it's the small intestine is very complicated. Really, the whole GI tract is. So what I want you to gather from this is that it's loaded with neurons. It has a very specialized set of kind of hormones that it could release through the endocrine pathway that we'll talk about, and one of them was cholecystokinin. And it also has, it's highly vascularized with blood supply. Okay, so things can travel into the blood and then travel to the stomach or the pancreas or the liver. Okay, the hormones can travel, right, and then signal something. Food could be absorbed into the blood vessel. Drugs can be absorbed into the blood vessels, right, and then they go through the portal vein to the liver. So we have a lot of stuff going on here. So a little bit more on the enteric nervous system. That is, again, considered like a second brain. There's about 100 million neurons in the gut, in the whole GI tract, about 100 million neurons. Okay, they are connected to the autonomic nervous system. However, some of them function independently of the autonomic nervous system. And what I mean by that is, if someone actually didn't have any innervation from the vagus nerve, from the parasympathetic nervous system, if that were cut or removed, but you still had a viable GI tract that were still functioning otherwise, you would still have the enteric nervous system doing a lot of things. So a lot of that is automated. So in the sense that if you have a bolus of food in the, or chyme in the small intestine, it can move on its own without the parasympathetic nervous system. There's some local regulation there that moves the musculature in the circular and longitudinal muscle there in those nerve plexuses. So again, there's a lot of independent functions. That's the enteric nervous system. The enteric nervous system has sensory functions in efferent or motor neurons. Okay, so there's efferent neurons and motor neurons, and they're also all connected to the central nervous system. So it's not really my goal to describe to you how every neuron communicates, from the small intestine to the brain and from the brain to the small intestine. And I just want you to understand that they're all interconnected, and that a signal coming from the brain can reach the enteric nervous system. A signal coming from the enteric nervous system can reach the brain, but a signal going to the enteric nervous system from the lumen of the intestines can also just come back onto the intestines for a motor response. Okay, so this is my really, really poor way of illustrating that. This is an actual nerve, a myenteric plexus, I think, from a cat. It's a real image from a dissected cat that I found online. So it's very different, right? Maybe different from humans, but just to show you what it really looks like, it's not nearly as uniform as you might expect from textbooks. the images of neurons. Okay, it looks very different, kind of just like a net. Okay, like a mesh of different sizes. But I'm just illustrating that every both plexuses are connected to the central nervous system. That would be sympathetic nervous system and parasympathetic nervous system. So both branches of the autonomic nervous system. So what that means is there's a reason there, right? So there's a reason why sympathetic and parasympathetic neurons are innervating the GI tract. Okay, and that is because they can control its motor and secretions. So that means if you need to slow down your gastric motility, perhaps you are exercising, you're running, okay, or classical example, you're escaping from a wild animal and you're a caveman, right? So you're trying to get away. The sympathetic nervous system will be activated, right, while you're running. Adrenaline will be released, or epinephrine, and that will actually trigger this GI tract to slow down. The gastric and intestinal motility will slow down, whereas the parasympathetic nervous system does the total opposite. Okay, so more parasympathetic activity means more gastric motility and more gastric secretions. So as you can imagine, we might have already talked about a number of drugs, in physiology, alluded to a few drugs that affect these sympathetic and parasympathetic nervous systems. And I know you haven't had pharmacotherapy yet, but when you think about it, or when you learn about it, you'll realize that there's just a large kind of array of drugs that can either block parasympathetic or stimulate parasympathetic, block sympathetic, stimulate sympathetic, essentially saying many drugs have side effects related to the GI tract because of the autonomic nervous system. One of the obvious ones is opioids. Okay, opioids are, it's a little more complicated, and we'll get into that later, but the opioids actually affect a neuron that's in between. It's called an interneuron. It's not at the beginning of the pathway or the end of the pathway. They actually block cholinergic transmission at one point. Okay, so that's somewhere kind of in the middle of the pathway. Whereas if someone happened to be exposed to I always mention this because I teach toxicology, but an organophosphate insecticide, those are acetylcholinesterase inhibitors. So they increase acetylcholine, so they function like pro-parasympathetic. So if someone were exposed to too much, or they had an overdose of an organophosphate insecticide, they would have hyperbowel secretions, hyperbowel motility. Okay, that can be diagnosed. Not too hard with the stethoscope, looking at bowel motility, and that's one way to figure out was that a parasympathetic or sympathetic toxin that they had. Okay, so just a reminder, in the intrinsic enteric nervous system, I just want to remind you that the main nerve plexuses again would be myenteric plexus, intervenes the longitudinal and circular muscle, as well as some mucosal glands. The submucosal plexus I would think of as less involved in muscular activity and more involved in secretions. Okay, even though it does innervate the muscularis mucosae, I want you to understand mostly that the submucosal plexus affects secretion of various substances by glands, basically glands there in the small intestine. Okay, it affects blood vessels, vasodilation, vasoconstriction. Please understand that the submucosal plexus it's more affecting blood supply, fluid supply, and secretions rather than the larger circular muscle and the longitudinal muscle. Okay, again, just to reiterate, the intrinsic afferent and efferent nerve fibers, they function regardless of autonomic input and are present in all sites. Okay, and again, just to clarify, intrinsic means that is embedded in the wall of the GI tract, right? That functions It has some function without central nervous system activity. So that's called the intrinsic enteric nervous system. Okay, so it has its own kind of local effect. So it has afferent and efferent nerve fibers. Again, and they pretty much can respond to the CNS, but they can also function on their own. Of all the neurotransmitters, that, believe it or not, there are a lot of different neurotransmitters involved in the enteric nervous system, the intrinsic enteric nervous system. One of the most important ones is actually 5-hydroxytryptamine. Okay, that's actually serotonin. So some people confuse that as 5-hydroxytryptophan, but it's actually 5-hydroxytryptamine, or 5-HT. I want to clarify, this is usually a point of confusion where 5-HT is actually serotonin, whereas 5-HT3 is considered the serotonin receptor, or one of the serotonin receptors. So again, just to clarify that, there are several different serotonin receptors. One of them is 5-HT3. If I just say 5-HT, that means actual serotonin. So even though a lot of people think of serotonin as primarily in the brain, affecting mood and other things, usually it's associated with a happy disposition or a positive mood, but it has a lot of other effects in the brain. But it also has a major role in the small intestines and in the GI tract in general. And it's been estimated that about 80%, literally 80% of the body's serotonin is in the GI tract. Okay, so there's a major role there. And it directly responds to nutrients and toxins and things. So I'll just talk about some of those things that it does, especially just the physiology of it, rather than spending a lot of time on the response to toxins. So let me back up for a second. Sorry about that. Yeah, so I forgot a little bit about the details here, but the next slide is really a continuation of this. So I just want to reiterate or clarify that 5-HT is released from enterochromaffin cells. I want to clarify, first of all, that is different from the ECL cells in the stomach. Those are enterochromaffin-like cells. And we're not talking about the stomach here, we're still talking about the small intestine. So 5-HT is released from something else called the enterochromaffin cells. Those are actually the apical membrane that is exposed to the contents in the lumen of the small intestine. So this 5-HT is actually released from those cells which are the enterochromaffin cells, which are right on the lumen of the GI tract in the small intestine. So if nutrients interact with they're going to have their own receptors, some of the nutrients. We have receptors on those enterochromaffin cells. If the nutrients react with them, or they physically or mechanically stretch the intestines will actually release serotonin. Serotonin will travel behind, not in the lumen, but it'll be behind the cell on the basolateral side. It'll leave there and find a neuron with a 5-HT3 receptor that would be considered a sensory or afferent neuron, and that would innervate either the myenteric or submucosal plexus. And this is just a continuation. The myenteric plexus would then trigger peristalsis through efferent innervation of the circular muscle. Okay, so it's actually hardwired, and I'll show you in a little bit a diagram about that. So wherever the food bolus is, the neurons are actually hardwired to contract before the bolus and to relax after the bolus, and that allows. When I say before and after, I mean before is closer to the upper GI tract, and after is closer to leaving the body, so closer to into the caudal direction. That's again caudal, towards the anus. That's basically what's happening. Okay, so you have to relax after the food, contract before the food, and then it'll move it forward. Okay, that is all hardwired. It's not by chance. It's not random. the neurons are wired that way, and we'll talk about that in a little bit. Okay, that's dependent on, particularly on cholinergic neurons, which are important for the contraction. And then the relaxation in front of, or I want to clarify what I mean by in front of, is where the food is supposed to be going, what direction it's supposed to be moving in, right? That front there is going to be relaxing, and that's through, believe it or not, nitric oxide, which is something you may not know much about yet, but we talked about it in the cardiovascular system. And nitric oxide is very potent at relaxing smooth muscle. Well, the same thing applies in not only in the vascular smooth muscle, but in the gastrointestinal smooth muscle. Nitric oxide is actually it's not just a signaling or a messenger. It is actually a neurotransmitter in some cases, right? It doesn't have to be a neurotransmitter, but because it can be released by other mechanisms. But some neurons do possess it. Okay, so nitric oxide and something called vasoactive intestinal peptide neurons cause relaxation. Okay, and there's a lot of other neurons, and the ones I'm really alluding to are the opioid neurons that we'll talk about, and because you have endogenous opioids or enkephalins that we'll talk about there. So the last bullet there is just to help you understand. So we actually have drugs that do this. We have 5-HT3 receptor antagonists. So when we use those, they actually decrease motility and increase water absorption, because if you decrease the very first step in this whole pathway, which is serotonin binding to its receptor in the afferent pathway, then you terminate the entire pathway and you decrease motility. Okay, so again, this is what I meant. So what should be happening? If there's a food bolus there, we should have contraction kind of like behind it, and then they're not showing it here actually, but there should be relaxation in front of it. Okay, but there's contraction behind it, and then it's going to move hopefully in the correct direction. Okay, so this is very simplified. So I kind of have a really almost embarrassing animation, but hopefully it helps a little bit. So this is just illustrating how the small intestine will respond to food. So imagine you have, and hopefully it won't be a full burger in your intestines, right? It should be at that point it should really be very, very small molecules, peptides and things like that. The further you get, so this would have been broken down into very small molecules and things. So basically, this blue ball is supposed to represent a food molecule, nutrient traveling to, this is the enterochromaffin cell. The purple balls represent serotonin or 5-HT. So when this enterochromaffin cell is activated by a nutrient, the 5-HT will leave behind the cell, travel to a neuron that's embedded in the wall. Okay, that's a serotonergic, well, I should say it has serotonin receptors on it, 5-HT3 receptors, which is the little purple thing here, purple receptor, that will send a signal, and this is what I was talking about, this how it's quote-unquote hardwired. So just imagine if the food is coming this way, down, the nutrients are coming down, we want to contract here above and relax below. And the way the body does that is by sending a signal up. And it starts off, it's not, this is pretty common in the body. It'll have like interneurons that are cholinergic, but then the final outcome is in many cases different. The final neuron is different. So if cholinergic up, cholinergic down, it's going. Acetylcholine is released here, but then that is released onto another cholinergic neuron. Okay, so this is really an interneuron. So it's cholinergic, and that causes constriction or contraction. That's above the food. Below it, okay, we're going below. We have a different type of neuron, that is the nitric oxide and vasoactive intestinal peptide releasing neuron. Both of those are I almost said vasodilatory, but I meant dilating intestinal smooth muscle, because we've been talking about cardio for many, many weeks. So I've been using vasodilation, but basically we're dilating the intestinal smooth muscle to allow the food to keep moving down, and we're pushing it up top. And I don't have a real good animation there, but just showing the kind of the food moving down. So the reason why that's vibrating is because I wanted to illustrate that this could be through chemical means or agitation or pressure like distension can trigger the serotonin release from the enterochromaffin cells. Okay, when this contracts, this relaxes, the food will continue moving. And this is a really terrible animation of contraction there. Okay, so that's it. So why does that continue to work? So if you move down further, the food keeps moving, the same thing is going to happen, right? We have another neuron that'll do the same thing. Constriction above, dilation below. Constriction above, dilation below, until it gets finally to the end of the pathway, right? And then you have defecation. So hopefully by the time that happens, right, most of the food has been absorbed, if not all of it, and then you'll have waste, right? So that's why the jejunum is so long. So this is happening throughout the entire intestine. So you can imagine probably that if any one of these were blocked, right, if we block this up here, that would terminate contraction. If we block this down here, that would terminate relaxation. Either way, if these processes are halted, we don't have peristalsis. Okay, and that's basically what this whole thing was here, is peristalsis. And I wanted to connect that whole concept to something that we really see. And again, I alluded to this earlier. What may be considered one of the most bothersome or nuisance side effects of opiate drugs, technically I guess it's opioid drugs, whereas opiates are natural, opioids are synthetic. Okay, but what would be one of the most nuisance side effects from OxyContin, morphine, hydrocodone, codeine, or even heroin? And the answer, if you know anyone who's taken these, you might know, but it's constipation. And that's because opioids actually inhibit a portion of this pathway that we just talked about, the neuronal transmission. Okay, and that's coming up now. Actually, we have something called an interneuron. So interneurons are in the myenteric plexus that influence the contraction. We have opioid interneurons. Your endogenous opioids are called enkephalins. They inhibit acetylcholine release and inhibit contraction. Okay, so I mentioned that that's a side effect, but as we often see in pharmacy here in the pharmacy field or therapeutics, sometimes we take advantage of side effects, use them as therapeutics. So that's why we have drugs like Imodium or loperamide. Loperamide is an opioid, but it does not cross the blood-brain barrier very well. So it has very little central effects. It doesn't have analgesia. You don't have analgesia, right? So it's not going to give you that high or analgesic effect. But because it does work peripherally in the small intestines, loperamide can actually treat diarrhea. rather than worrying about its side effect of constipation. That's not even relevant, right? Because it's actually used to treat diarrhea. So we're hoping that it reduces bowel motility and gives more time for absorption of fluids. Okay, and I'm not going to focus much on these, but dopamine-releasing neurons are also inhibitory, and you'll learn about those when I talk about nausea and vomiting in the gastrointestinal module. Okay, so what do I mean by that? So let's back up and remind you of this diagram. Okay, in this diagram, we're going to go up the top here. We have a cholinergic neuron and another cholinergic neuron. Okay, and that would promote contraction. However, So again, cholinergic neuron, the same part of that pathway there. However, there are a lot of regulatory neurons or interneurons. Okay, some of them are dopaminergic, some of them are enkephalin releasing or opioids that we just talked about. So what I want you to understand from this is that, so this negative, first of all, that means it's inhibiting, I want you to understand that means it's inhibiting this neuron. Okay, that negative there means that neuron is inhibiting the cholinergic neuron. So, and that does that through releasing natural enkephalins. And when they bind to their opioid receptor, one of them is the mu opioid receptor, it's pretty common. Um, it's activating the receptor, but activating the receptor shuts this neuron off. It slows down the cholinergic neuron, and then you have less acetylcholine at the neuromuscular junction, and then you have less contraction. Okay, so that means if we give a drug that eventually it can diffuse from the capillaries, it can diffuse to the same location, and any opioid drug will work here as well, causing either constipation, or if it's a therapeutic opioid, right, we can treat diarrhea, right? So in many cases, though, the opioids are used just for pain, and then that's a side effect, is constipation, which can be potentially really dangerous in someone who, for instance, someone who's had a stroke and they already have perturbed or abnormal innervation in their GI tract, and they already cannot produce a normal bowel movement. They may already be prone to constipation, and then if they have pain and they get an opioid, it's going to make that even worse. So certain patients are even more susceptible to that constipation from opioids. So it could be a lot more than just a nuisance. It could be a pretty severe side effect in some patients. So the reason why I brought that up now is because we're going to talk about opioids in the curriculum many times, and just understanding that, again, this is still physiology, because enkephalin neurons are still, they're endogenous, they're neurons. Enkephalins are natural, so this is physiology, but we can understand the drugs very easily just by understanding this physiology. So again, too much opioid, too little acetylcholine, and then this whole contraction here just stops, right? And then we have no bowel motility. And why does that matter? Well, it means not only is not only is the bolus of food stopping, but it gives more time for water absorption, and then it gets desiccated. Okay, this next slide I think is a little bit more detail than I should have, just for the physiology course. So, I had this in here for a few years because I do teach nausea and vomiting later. So I wanted to like kind of give you this precursor to that. But I think it's a little bit too much detail. Just, if you're interested, you can read it. I'm not going to ask you test questions on this. But what this is kind of illustrating is that, again, this is in the small intestine as well. These are the enterochromaffin cells that possess serotonin, or 5-HT. I want you to understand, again, FYI for now, but when these get, remember, when food interacts with these, that's a kind of a productive stimulus. However, if there's a toxin that actually damages these cells, believe it or not, if they release enough serotonin, that could actually be, an overkill, and too much serotonin that can actually cause pain. And one of the things that not only pain is, it can cause nausea and vomiting. So too much serotonin can trigger to the vagal afferent neuron to the brainstem. If there's an overabundance of serotonin hitting this, that can actually trigger nausea and vomiting. So it's interesting. The same receptor is what is sensing nutrients to trigger things like satiety, right? You have food there, so it might trigger your satiety reflex to stop eating if there's enough food there. However, if you overstimulate this by like literally destroying this cell, having excess serotonin release, that will cause nausea and vomiting. And then another one would be, again, toxins here can directly cause pain through release of something called substance P, which signals to the central nervous system. Again, not too much, nothing to memorize there, just something I thought was kind of interesting. Whoops, a little animation. Electricity, that means excess stimulation. I will stop here and then continue with absorption in the small intestine next. Some pretty simple stuff, and then I will get into lipid homeostasis because it's so important for pharmacists to understand that, even though that's in a different chapter. Spend some time on that.